start by taking a couple of seconds to congratulate Connie and the Angioma Alliance Canada for this amazing meeting, hybrid meeting that kind of brings all of us together. And every time I think, the next one can't be better, we've proven wrong again. So it's been a great meeting. I learned a lot, and hopefully we can spend the next few minutes picking each other's brain as to what are the things that are most pressing in your minds about what you heard during this meeting. And I will try to do what I can to kind of translate it into doctoring 101, just the basic thing you want to hear from your doctor, and we'll try to do that. So I don't know how you want to start that. I did not prepare very specific remarks. If you want a summary, we can do that. If we want the questions and answers to drive it. Connie, what do you have in mind? We can do whatever way you want.
We'll do an interview.
Interview is good.
Okay, we'll start. So, yes, please tell us what you thought the highlights of the scientific meeting were and what brings you the most hope out of what you heard today and yesterday.
Yeah, so highlights, in my view, fit into two categories. What we're doing best now and what's exciting breaking out. But the other highlights in my mind that excite me even more is new ideas that are going to play out over the next few years. So in terms of what's really now playing out, I think we're right in the mud of analyzing the first meaningful clinical trials, and I think it's going to be a very exciting thing. And I've been doing clinical trials and other diseases for about 20 years or so, and I want to just tell you that you learn as much from a negative trial as you do from a positive trial. And almost every time we do a trial, the first trial will be negative, or the first 2 or 3 trials. What negative means is not that it's bad. What negative means is it was neutral on what we call the null hypothesis, which is what we wanted to try to test. But the trial generates high quality data that otherwise is unavailable. So let me give you just a very quick example of that. We could putz trying a medicine in one person or another for 10, 15 years, and I think it works until it doesn't work, and I think it's working for me unless my cousin tells me it didn't work for her, and whatever. We'll never get to the truth. Yet you use that time in a controlled analysis where a third party is looking at the outcome, not the biased parties, and then some amazing truths come up that teach us how to ask the question better, either the same drug asking the question better or asking it differently, and so forth. And we are going to get positive results no matter what, because there will be signals that motivate the next hypothesis. I think the most exciting, the way I see it, that's in the next few months is going to break right out, and you've heard a lot about it now, is the results of trials that are playing out and biomarkers work that is being developed and so forth, and maybe that will drive some Q&As. But the other part that really, there are a few things in the history of this meeting over the last 20 plus years that were like aha moments. The first time I remember walking out and saying, this is really going to change, is when Angela left, but it was Angela's work on endothelial cells in Dr. Ginsberg's lab. And when she inhibited the CCM gene, she showed that the endothelium no longer formed tubes, but it forms bubbles. And it's so fundamental, it's in the dish, but it was an aha moment that for the next 10 years we figured out the rock hypothesis, and those are now the drugs being tested in trials. So the aha moment, I think, at this meeting is focused and targeted delivery. I think there are going to be a lot of things that we think work, but we don't want them to go to the wrong endothelium. We don't want the dose to be wrong. Rapamycin is a perfect example. If we give it in the wrong way, but maybe doing more harm than good. Yet if you give it in the right way, it could do something very dramatic. So we have to this focused, targeted recovery. Delivery, and it's not how we initially think. It may not be targeting endothelium, because another person will tell you, well, endothelium is open to the bloodstream, so it may be improving perfusion like the ultrasound people are doing. They think they're doing permeability, they may be doing perfusion and they don't know it, and even gene therapy that we're testing now, how to deliver a gene, and Andrew, I want to commend him on trying to put these concepts in an understandable way. He's very, very good at that. And the idea of delivering a gene, you can't get it into the wrong cells. It's got to go to the target, and eventually we're going to show a proof of concept in the mouse, but that's not how we're going to do it in the human. The human has to be more targeted. So I think, hopefully we can get in the next couple of meetings real experts on focused targeted delivery. We are kind of amateurs at it. We know CCM more than them, but we don't know what's really happening in the nanotechnology world. I mean, some people are really doing a lot of stuff, and we need to put our minds together, learn from them, and they learn what we need in our disease. So I think those are the things. Sorry about the lengthy answer, but that's kind of what. Come on, you guys are not shy. What is it?
Explain to us what a biomarker is and why biomarkers are important.
Great, thank you. I love that question. So a biomarker starts by being a biologic signal of a disease state. So I'm going to give you some examples of biomarkers you know about all the time. So hemoglobin A1C is a biomarker of diabetes. So it works as a diagnostic biomarker. So if you have it above 6, you are a diabetic. If it is, above 5.6, you're a pre-diabetic. So it diagnoses, so it's a diagnostic biomarker. It is also a prognostic biomarker. So if it is higher than 7 or 8, you start having cardiovascular complications of diabetes, renal failure of diabetes. So it is a prognostic biomarker. It is also a monitoring biomarker. So you take metformin or you take Ozempic or whatever, and it tells you that that drug is working on your diabetes. So you've just heard about the 3 contexts of use, right? So a diagnostic biomarker is one that's used to diagnose a question in the disease. A prognostic biomarker is to predict an outcome, and a monitoring biomarker is to figure out if what you're doing is right or wrong for the disease. So in CCM, I know you don't want to hear about diabetes, we want to talk about CCM. So in CCM, we struggled together, but the patient community helped inform us, and my Monday clinic, for those who see me in Chicago, that's kind of my education. That's my classroom, because I see about 20 CCM patients every Monday, and that's how I learn what you need and what you want. So in those clinics, what I heard is an important question: is my cavernoma unstable? Is my headache in the last 6 months meant that I had little bleeds in my cavernoma? And maybe I did the MRI too late, maybe I didn't do an MRI in time, and I missed the chance to know if I had a bleed or I didn't have a bleed. And you can't every day have an MRI. So I think there is a very good context of use for a diagnostic biomarker of the lesion that bled in the prior year. So if you have an MRI and you see the blood, you don't need the blood test. But many don't have that, and many have the question, that is my innocuous lesion, is it actually fooling me? And it's not as innocuous. In fact, I've been having these weird headaches, I don't feel right, is it leaking or not? So we worked very hard to develop a blood test that separates the patients whose CCM we know bled from those who we know did not bleed. So those that had an MRI that proved no bleed, and those that had an MRI that did prove bleed are SH, no SH, cash, no cash. And sure enough, their blood tests are different, and they're different in ways that you can't know what to look for, because blood tests for this type of question is like a needle in a haystack. There are millions, gazillions, whatever the number of molecules in the blood, and if you know what you're looking for, you're gonna discover the right molecule to go after. And we did a lot of that through mechanism studies, through mouse studies, and through what we call unsupervised discovery, meaning taking the two groups and see what shows up between them. Doing all of this together, we found that in fact the blood test can be close to 90% accurate, separating the bleed from non-bleed lesions in diagnostic context of use. So asking the question backward, did my lesion bleed? And it'll never be 100% because we're never sure that the non-bleed didn't really be non-bleed, right? So what we call biologic truth is only true when you have a bleed. It's not so true when you don't have a bleed. So we'll get close to 90%, but that's going to be very good, and the FDA knows that, and they're excited about it. So what do they want from us now? We found several combinations of molecules that could do that. It'll be one of those composite tests. Has anybody heard of the concussion blood test? So there is a blood test that is developed by the Abbott laboratory, and it's developed so that when you're on the field or after a head injury, or for the U.S. Army, whether you know, a bomb went out near you, whether you were concussed or not. And it's made up of two proteins. One of them is a glial activation protein, and one of them is an inflammation protein. And the combination of those two proteins will tell you if you have had a concussion, and the severity of it tells you whether you should go to the hospital. And they developed it to the point that they can do it on a pinprick test in the field, so that the idea would be, we'll fly out the soldier off to base if they have more than 30,000 GFAP, you know, and the same for the field, whether you go back to play ball or not. So it's similar to that, but in our area, it's going to be 3 to 4 molecules. It's not that far off. You know, so 3 to 4 molecules. There are several combinations that work. So what the FDA wants from us is to know that it works in Chicago at Mayo Clinic and in San Francisco and Barrow the same way. So they want multi-site validation. They also want analytic validation, meaning the measure I get in my test is the same that could be reproduced if they ran the test at Duke or they ran the test somewhere else. And they want to know that it applies to all the subgroups of the disease. So does it work the same in men and women? Does it work the same in the young and old? And what might be confounders? If you're an epileptic, does that make the test not work or work? So we had to assemble about 800 patients to answer that question, because each subgroup has to be about 40-40. And that's what the biomarker project is, 800 patients, what it takes to do all of that. And we're about 4/5 of the way through that project. So it's really has been going great. And at the end of it, I expect us that we will go with what we call a letter of intent to the FDA, and letting them know that that is what we want to have them approve. And it's at that time that we will partner with a company, whether it will be Abbott or someone, that will will take interest in developing, just like pharma companies have stepped in to test drugs. So that's kind of when it goes from the academy to the market. So estimate of when that diagnostic test will be useful and in your hands, probably as short as 5 years and as long as 7 or 8. So it's not that far out. And for the familial disease and for those young among you, it's going to affect you, so it's going to be great. And that's kind of the diagnostic context. The prognostic context is even more exciting because that one is the one where I take your test today and I can predict whether your lesion is likely to bleed or grow or stay stable. Okay? Who doesn't want to know that? Can somebody just raise your hand if you don't care to know that? Okay, everybody wants to know that. And in fact, the accuracy of the test is higher in the prognostic context because we are able to know the biologic truth for sure, so we not separate it. The only downside is that the frequency of events is lower, so it's taking us a lot longer to collect enough patients that have had the bleed to compare them to those that didn't. So for everyone I see, and thank God, only 1 out of 15 or 20 bleed next year. And that's fine. I don't want it more than that. But that means I have a lot more patients to develop and whatever. So that test will definitely be in the books, but it's going to be probably 3 to 4 years later than the diagnostic test, because of these practicalities. So we will be able to go to the FDA with a letter of intent about three years or so after the other one. So it'll be that much delayed. Now, having established the first and maybe gotten a company interested will accelerate the development of the product. And the third context of use is the monitoring biomarker, and I presented a paper on that yesterday. And all 3 drug companies that are interested in this now want to apply it, okay? So what that test means is that if your lesion is bleeding and you're giving it a drug to decrease the bleeding, can we measure that with a blood test? So the change in your blood test. And we have already the formula for almost perfect test for that. And that one is hard to develop for the public because, until we have a drug that's widely used, who's going to use it? I mean, how do you know to apply it? But the drug companies have a lot of interest in it, and they have millions of dollars to invest because it will cut shorter their trial costs. So they're able to monitor the effect of their drug and go to the FDA and whatever. So with each of the 3 companies, we're trying to put our heads together. And I decided long ago at the advice of Connie that there is no exclusivity in this. We're open partners. So you want me to help you, I'll help you, but that doesn't mean I'm not helping your competitors. So I'm working with everybody. And the idea here is whoever wants to move it, let's move it. Let's let the competition play out, and we will let them use that test and our discovery in their drug development. And those are the 3 contexts of use for biomarkers for CCM. Long answer to a short question, but that was our intent. You wanted to start talking. All right.
Sometimes the doctors say, watch and wait, as opposed to surgery. With all your experience, can you give us some reassurance that, or some words of wisdom about watch and wait versus surgery? When should we be saying, no, I'm not happy with watch and wait, I want a second opinion, I think I need surgery? Can you give us
Yes,
advice?
so we have guidelines for that there is actually almost near perfect agreement about among surgeons currently and non-surgeons. So when you put neurologists and neurosurgeons together, they have a number of criteria under which they can give you recommendations with what we call high level of confidence and high class of evidence, meaning the evidence is not just I think so. There is evidence for it, controlled evidence. So what do we know? So we know that if your lesion has actively bled and it is in a safe area in the brain to be taken out, it's probably better to take it out. Now, the issues here is that what's safe, right? I mean, if you're, there's no brain surgery that's 100% safe. So you have to kind of look at that. There is the other issue. There are some spots in the brain where the price of removing it is worse than the bleed. And therefore, you may decide, I want to wait for 2 bleeds before I recommend surgery in that area. And there are other places where even 4 bleeds, not good to do surgery, So the answer will not be yes for everything, So the devil is in the details, and it depends on the lesion and how it's behaved and how it's likely to behave. The good news is that we know that a lesion that has never bled, that has been stable, that is discovered by chance, you're more likely to harm it by taking it out. But once it starts to misbehave, it changes the equation. So yeah, I had a lesion discovered because I had an MRI to make sure I don't have a met from my breast cancer or whatever. That lesion is different than the lesion that is discovered with new blood because I've just had a seizure, right? So they're different, and I think we have evidence not to do in the quiet lesions that have not caused symptoms. And we have evidence not to do if the surgery causes more harm than the bleed. And we have evidence to do it and not to delay in the other situations. I want to just tell you a scenario in which I try to twist the patient's hand to have surgery. And those who know me know I don't do that very often. But I do that if, for example, an 18-year-old has a single lesion and they just had a focal bleed in that lesion and the first seizure in their life. Okay? So they were perfect, living their life. They just had a seizure. We see blood. We know that the seizure is caused by this lesion. That patient, I got to take this lesion out. Because first of all, they will never bleed again. Second, if it's a solitary lesion, they're cured from the cavernoma forever and ever. And the third reason is that they're likely to be able to be taken off the seizure medicine. Okay? So those 3 are very compelling. I'm going to twist your hand to get it out. But it's not the case if you have multiple lesions and your seizure, we're not sure which lesion it's coming from, then you start doing more harm by doing the same thing for that patient. Does that help how we approach it? No problem.
Thank you, Dr. Awad. My question is actually bringing it back to the role of bleed and what a bleed actually is. So, we have been told over the years that Rock doesn't have no bleed, but we know that
of course he's had bleed.
Yeah, so that's really my question. I suppose there seems to be maybe uncertainty around what bleed is and bleed isn't.
There isn't really, because every cavernoma has bled by definition. Okay, so the cavernoma is actually a lesion that has blood in it. Therefore, at some point, it has bled. Okay? However, there is a huge difference between a cavernoma that has fresh blood with new symptoms and ones that doesn't. Now, of course, if you don't have a cavernoma, you're not going to have a bleed from a cavernoma. So I want exactly what Connie is saying. I don't want to have a cavernoma. You have the gene. I don't want to have 10 lesions to watch. We don't want to have 10 lesions at all. But today we're stuck, right? So we have 1 to 10 to 20, whatever. So then you have to separate the ones that have active bleeding with new symptoms at the time of the active bleeding, because that separates a subgroup of lesions that merit a more urgent intervention like surgery and can make a difference in the patient's life. So that doesn't mean the others are not important or they didn't bleed, but it separates these, what I call cash lesion, because I think that's where the money is today. That doesn't mean that's not where the money is, the other lesions, but in fact, it is very important. It's a tenfold difference. So let's take a situation that you're familiar with. I've had 20 lesions. And then every time I do a scan, none of them has changed, versus one where one of them starts to change and now the seizures change. That warrants separating it from the others. That doesn't mean the others are okay. And the others can disable you from epilepsy, can disable you cognitively, can disable you because of the anxiety of living and not knowing what's going to happen. We get it. But that doesn't mean they're all the same, because they're not. And we have actionable strategies for that group that misbehaves. Biologically, there are actionable strategies for these lesions that are separate from the others, because we now know, for example, they respond differently to rapamycin. So the growing big ones may be easier or more appropriate to target for that, while the background ones may be made worse potentially. So those are the types of things we have to learn from and weigh.
For more than a decade, and all of a sudden they develop focal epilepsy. It suddenly felt of accumulated bleeding over time, and often it just hit a threshold.
Yeah. So there are three scenarios where that happens, and we know about all those three scenarios. The question is that in the case you're asking, I don't know because I don't know the details. But there is one scenario where all the lesions are sitting in different parts of the brain, but one is sitting in a part of the brain that is more vulnerable to epilepsy. So the medial part of the temporal lobe is one of them, the limbic system is one of them, So all the other lesions can leak and irritate, but nothing happens, or you get headaches or migraine. But that one, eventually that irritation triggers epilepsy. And it's more likely to trigger epilepsy if you're predisposed to epilepsy. So we have genes that if you have the same lesion in the same spot, one person gets seizures, the other never gets seizures. So that can't be a difference in the lesion, that's a difference in the host. Right? And there is a difference in the location. So epilepsy is complicated because of that. Then there is a second scenario where that lesion may have bled and you didn't know because you didn't do an MRI within a month or two of when it bled. You didn't have a reason to do an MRI, but it could have bled and then triggered epilepsy. And by the time you do the MRI after the second seizure or the first seizure, it may have been a month after the bleed, and the MRI doesn't show you the bleed. So that lesion is more unstable, but you didn't know it. And currently, we don't have very good markers to separate a lesion like that bled a few months ago, and we didn't detect it. Okay, so that's the second scenario. And the third scenario is a more overt one. And we see that all the time in familial cases, which is they get the seizure and one of the 20 lesions bled. You see it. That lesion is the epileptic lesion. If the EEG shows that, and then we can remove that lesion and cure the epilepsy, even if you're familial. So in those cases, we cure your epilepsy, even though we don't cure the CCM disease. So sorry, I hope I answered.